Good morning, ladies and gentlemen. It gives me immense pleasure in welcoming each one of you at this webinar titled Upstream Bioreactor Technology, Benchtop to Manufacturing, brought to you by Bluetech Media and powered by Paul Corporation. With the growing healthcare demands, especially seen in this pandemic, there are currently more vaccine and biotherapeutic manufacturers than before to step up to the crucial requirement of speed, scale, and supplies. With variety of bioreactors available in the market for development and manufacture of therapeutics. The major bottleneck to commercialization is the viability of a cell culture process in addressing scale-up and technology transfer. This webinar outlines the aspect of bioreactor designs, characterizations, and scale-up of a single-use stirred tank bioreactor and highlight the benefits that an advanced flexible control system can bring to bench scale bioreactors catering to both adherent and suspension cell culture processes. To start the webinar today, let me welcome our guest speaker, Mr. Satish Kagindali. He is the Senior Scientist, Upstream Vaccines R&D, Reliance Life Sciences, to share his views on single-use bioreactors and process optimization challenges. Satish is a biotechnology professional with a vast experience in bacterial, yeast, and cell culture upstream R&D scale-up, process development, and technology transfer, handling QMS and troubleshooting related to process and equipments, preparation of URS, FAT, SAT, IO, OQ, and PQ area and utility qualifications. So over to you, Satish.
Thank you, Ashish, for the introduction. Hello, good morning, welcome you all. Today I'm going to present a small presentation on bioreactors and challenges. So let me start my presentation by sharing my screen. Okay. Again, good morning all. Today we'll discuss about bioreactors and challenges. So let's get started. Yeah, these are the contents of my today's topic: types of bioreactors, single-use bioreactors in suspension cell culture, single-use bioreactors for in adherent cell culture, and challenges in conventional bioreactors and single-use bioreactors, and a couple of slides on utility and time management. That is at the 2 kL capacity. So bioreactors. These SS bioreactors are derived from fermenters. It's single bioreactors are introduced 25 years back. Single-use bioreactors are now disposable bags, which consists of 3 layers of plastic foils made up of plastic foils. So before single-use bioreactors, to conduct clone screening and media screening activities, we used microplates and shake flasks and all. We used to select like 3 or 4 clones and carried out into glass bench bioreactors. And this resulted in low yield due to lack of mimicking of this shake flask microplates to bioreactors, because we don't control so many parameters in microplates and shake flask. So due to which we ended up in low yield clones. So now with single-use bioreactors, we can run 96 experiments or even more than that simultaneously, and and results can be analyzed using DOI tools. We can analyze the results also within weeks. So previously, the average titers used to be 1 to 1.5 gram per liter. This is regards to monoclonal antibodies. Now, with the help of this, new technology, which is single-use bioreactors, we can say that the average has moved from 1.1 to 1.5 gram per liter to it's almost double now. It's 3 gram per liter. So this is a main advantage of using a single bioreactors. So coming to types of bioreactors. So as you all know, the SS state tank bioreactors are having application, wide application in suspension cell culture as well as adherent cell culture. Scale ranging from 20 to 20 liter to 20 KL. Suspension cell culture application, we can use it to as well as for adherent also by using microcarriers. So beads can be added into the stir tank, and we can use it for adherent cell culture process. Again, stir tank single-use bioreactors. These mimic SS bioreactors. So almost mimic the SS bioreactors. Again, these are pre-layered plastic bags ranging from 50 liter to 2 KL for suspension cell culture, and these have different shapes and sizes, like cylindrical as well as cubical shapes. Cylindrical usually consumes a lot of space in terms of height. That again, cubical you can reduce the space in terms of height. Height by H by D ratio it will be 1:1 in terms of cubical shape bioreactors. Next is wave-induced single-use bioreactors. They are usually 2D and 3D bags. Usually these are used for seed development activities. These range from 1 liter to 200 liter scale. Usually seed developmental activities, these can be used. Next, coming to dynamic bed single-use bioreactor, these are used for adherent cell culture activity. Usually, these will be in disc-shaped microcarriers. Usually, that is not the compulsion, and these are usually made up of polyester microfibers, and these provides larger surface area when compared to microcarriers, as in case of stir tank SS bioreactors, where we use beads to carry out adherent cell culture. Dynamic bed reactors will offer a much more surface area compared to microcarrier technology. Moving to next slides. These are some of the single-use bioreactors available in the market for suspension cell culture. First, we have PAL. Allegro XRS 25 bags are available. This is a unique technology, a biaxial agitation system, which shorter the mixing duration from 50 to 90 seconds. In case of conventional system, it will reduce it to 16 seconds, and these ranges from 2 liter to 25 liter scale. And again, in PAL we have Allegro SPR bioreactors, which range from 50 liter to 2 KL. In Merck, we have Mobius series bioreactors, which are usually disposable vessels starting from 3 liter to 2 KL. Again, in Appendaf, we have BioBlue, which is 380 liter to 50 liter scale. These are also plastic vessels. In Cytiva, we have Wave 25, which is used for seed generation activity, which is up to 25 liter scale. We have available in the market. Again, in Cytiva, for higher scale, we have XDR series ranging from 10 liter to 2 KL. So these are the some of the suspension cell culture single-use bioreactors available in the market. Coming to furthermore, like Sartorius, we have Amber series single-use bioreactors. Amber 15 and Amber 250 series are available. So these are widely used in the application of clone screening and initial media screening applications. So multiple parallelly we can run multiple batches using these Amber series bioreactors. Sartorius also offers biostat bags range from 50 liter to 2 kL, and also Sartorius offers biostat RM. These are the bag type reactors for wave bioreactors for seed culture application from 2 liter to 200 liter. Again, we have a biostat RMTX system, which is a unique fed batch and perfusion system for bags. And lastly, we have universal SU, which is again single-use tanks made of a plastic, which range from 0.6 liter to 2 liters. So in thermo, we have again high performer Rocker bioreactors. Again, these are the bags for seed development activity. Up to 50 liter working volume are available in the market. Again, in mid-range for pilot scale, we have high performer single-use bioreactors from 50 to 2 kL. Thermo also offers high performer dyno-drive single-use bioreactors, which is quite different. From 50 liters to 5 kL bags are available. So next, so these are some of the single-use bioreactors available for adherent cell culture process. So from PAL, Isolis single-use fixed bed reactor nano is available, which offers up to 4 meter square area. In mid-level for pilot scale, we have Isolis 500 plus system. This offers up to 500 meter square area. Again, in large scale, GMP model is also available in PAL. There is expansion multi-plate bioreactor system, which offers up to 1,224 meter square area, which is a huge area. Next segment is OmniBRX, which offers 0.5 liters to 200 liters vessels, in which we can get 1 meter square to 1,500 meter square area for adherent cell culture activity. So these are all the some of the bioreactors available for suspension as well as adherent. So some of the reactors I might have missed. So coming to challenges, challenges related to single-use bioreactors as well as conventional bioreactors. So which involves cost, safety, time, quality. So these aspects are the challenges which we face in single-use bioreactors as well as conventional bioreactor system. So advantages of single-use bioreactors over SS bioreactors. So single-use bioreactors eliminates cleaning validation process, as the cleaning is minimal. So we can reduce these activities like selection of disinfectants and CIP chemicals, preparation of protocols, approvals, execution of multiple cycles of CIP and the disinfection process. So this is a huge task and which consumes a lot of manpower as well as consumables as well as utilities. So we need to send the samples for CAP, and post CAP we need to send the samples and we need to wait for the results. So these can be avoided in single-use bioreactors, whereas there is very minimal cleaning activity required. Next one is the shorten downtime and turnaround time. So when it comes to subs offer downtime during post-harvest activities, which is usually a decontamination activity, which is very minimal. And turnaround time is the preparation activity for the next batch, which involves CAP, pressure test, ESAP, full vessel SIP, and thermocycle installation. All these activities, which require a lot of time and manpower and utilities and cost involved, these can be minimized. And subs offers low risk of cross contamination with every batch you are going to use a new batch, so which eliminates the risk of cross contamination. Again, as we have a lesser cleaning requirement, so we also have an advantage over minimal number of autoclaves and DHS load pattern qualification, as well as validation documents, qualification and pre-qualification process. Again, when it comes to capital cost and operating cost, more than 25% savings on startup capital can be saved using single-use bioreactors. And when it comes to utility and space, the cost goes down up to 50%, and reduced manpower cost considerably. And also the installation is very handy and very easy. As we are eliminating the SIP and CIP process, so it also eliminates the design of boilers, water purification systems, and steam lines. All these design activities also can be minimized. It also reduces the carbon footprint by 70%. You can see the example here. Paul expansion 200 bioreactor, which is hardly 80 centimeter height, which is equivalent to 20 multi-tray vessels. This is a huge amount of plastics which can be saved. That in turn reduces the carbon footprint. Again, now we are coming to another segment, challenges with the single-use bioreactors. So in single-use bioreactors, scalability is an issue. Like mass transfer is a challenge with higher scales. This topic will be handled by Mr. Pankaj. Then larger scale bioreactors handling is a challenge because of the area it occupies and handling without puncture and all those things. As during the installation, it also need to be air need to be passed inside the bioreactors to flatter it, and you need to check the integrity, all the ports, all these things we need to check. So this is a little hectic, and it consumes a lot amount of time and manpower. These are single-use bioreactors are expensive. Now, due to a pandemic situation, supply chain has in a bad situation. The price are almost double. Performance is not completely proven. This doesn't mean that single-use bioreactors are not well at performance. When it compared to conventional SS bioreactors, single-use bioreactors is a new technology, though it is introduced 25 years back. So we have limited data on single-use bioreactors when compared to conventional bioreactors, which are there from decades. So again, one more challenge is most of the large-scale facilities are already designed for with SS tanks and requirements for cleaning and all. So it is difficult to invest further on single-use bioreactor system. So coming to one more challenge with single-use bioreactor is leachable and extractable. So manufacturers will use stabilizers like this phosphate for stabilizing the three-layered bioreactors. So this under stress conditions like high temperature, irradiation conditions, it will lead to the chemical conversion of this phosphate into this phosphate, which is found to be detrimental for cell growth. Again, single-use bioreactors are potential for puncture. When you go for a higher scale, 1 kL to 2 kL, as you scale it up to 5 kL. So handling these bioreactors and installation without puncture is a difficult task. And these things are difficult to move when they are filled with media or culture. So they need some excess tank support to move in from one place to another. Again, these are sensitive for temperature and pressure. Temperature range is given for the most of the bioreactors as 40 degrees Celsius. When you can consider the worst case scenario, and if the temperature is high during the transportation and all, so this leads to chemical changes, and that leads to formation of this phosphate kind of things. And disposable cost also is high in single-use bioreactors, as the size of these are large. So next segment is I'm giving an example with the time and utility. So this is the mimic of 2 kL bioreactors run at the production scale. So this is the exact duration we have captured in the slide. So this involves revival, pre-seed 1, pre-seed 2, seed 1, seed 2, and seed bioreactor, and followed by production fermentation at 2 kL. So these involves first 3 stages involve 48 to 72 hours, which is common in conventional as well as single-use bioreactors. When it comes to single-use bioreactors at seed 1 and seed 2 level, we can reduce the time almost by 12 to 16 hours for each run. So you can say for every month you can run maybe 2 batches, because I'm considering here 300 hours batch. So 300 hours batch. So when in comparison with conventional bioreactors, it almost need another 2.5 days. So annually, if you consider, almost 1 month will be wasted in this process. So next segment is utility. As you can see, the process remains same. For single-use bioreactors, we have some common gases requirement. When it comes to conventional process, we require chiller, steam, chill water, steam, PW, WFI, alkaline acid. So these again, generators for these also required. So the lines to carry these steams and chill water and WFI. So these are all required when it comes to conventional process. These can be minimized in single-use bioreactors. So last slide summary. Of course, single-use bioreactors are economical and time-saving. So micro bioreactors or mini bioreactors will be helpful during the initial stage, like clone screening and media optimization, initial media optimization, to obtain a higher titer value. Hybrid mechanism can also be adopted, wherein if you already have an SS conventional bioreactor system, you can use it for production scale, and for seed generation, you can use these single-use bioreactors. And also, single-use bioreactors can be recycled, and also it will reduce the carbon footprint by 70%. So that's it. Thank you. I'd like to thank Bluetech Media and Paul for arranging this magnificent presentation. And I'll also thank my boss, Dr. Rajan, Sriraman, and Dr. Mohan for their support. Thank you.
Thank you, Satish, for such a thoughtful presentation. Before we proceed further, I would like to inform all our participants that we'll open the floor for a question and answers at the end of the session. You just need to click the Q&A button at the center of the screen below. Write your question by clicking Ask a Question, mention the name of the speaker, and write your question below. We will take this up with the respective speakers. Also, you will find feedback button. So please take a minute and give your valuable feedback before you leave. Now, it's time for a poll question. On your right-hand side, when you click Q&A button, you will get a poll button as well. Kindly click the poll question and you will find our first poll. The question is, what is the cell type you are working on? Options are option 1, adherent cell culture. Option 2, suspension cell culture. We will allow 30 seconds for you so that you can answer the poll question. Thank you very much. You can still keep the window open for the poll questions and we'll come back to our next poll in some time. Now for our next session, I would like to welcome Dr. Ratnesh Jain, Head and Course Coordinator, Bioprocess Technology, ICT, Mumbai. He will be sharing his views on the topic Digital Twin Next Generation Bioreactor Control Technology. Dr. Ratnesh Jain is a recipient of BIRSC Innovator Award, Humboldt Fellowship, and Ramalingaswamy Fellowship. Dr. Jain has demonstrated industry-academia collaboration and have completed more than 50 sponsored industry projects in the last 10 years. He is also founder of WeTranslate and co-founder of Avai Bioscience, a 3D printing startup jointly founded by IITM and ICT alumni. Dr. Jain is also leading skill development and training program and organizes one of the largest biopharmaceutical hands-on training program called Biosimilar Workshop. So over to you, Mr. Dr. Jain.
Thank you, Ashish. Thank you for the nice introduction. I also thank Bluetech Media and Paul Corporation for arranging this particular webinar. In few minutes, I'll be sharing my screen and we'll be talking on this particular subject, which is very much essential in the coming days, which we call Biopharma 4, or in a simple understanding, we call Industry 4. So Biopharma 4 or Industry 4, where digital twin is a very important part, is a next generation bioreactor control technology. Before I begin, I'll quickly introduce my institute, ICT Mumbai, which is set up in 1933. This particular institute is located in Matunga, Mumbai, and run one of the very important chemical engineering and very prestigious chemical engineering program, along with masters in pharmaceutical sciences, as well as biotechnology, or which we call bioprocess technology program, which is currently I'm heading. Also, this has unparalleled impact to Indian society, which has generated multiple chemical entrepreneurs So we host various alumni from ICT who gives a lot of motivation for us to work on bioprocessing area, starting from Reliance or Dr. Reddy's or Lupin and Marico. Coming back to our research group is basically divided into two parts. One is called Pharmaceutical Biotechnology Lab, another is called Biological Characterization Lab, led by myself. Looking into manufacturing biopharmaceuticals, so this is a primary process, and my previous speaker has already briefed you how bioreactor works. So this is an interesting concept, and for a very long time, we are working on biomanufacturing. So whether you are manufacturing an enzyme, whether you are manufacturing a recombinant protein, or whether you are manufacturing vaccines, largely bioreactors play an important role. So post clone selection, post media preparation, and post you understand that process can be scaled up from a laboratory scale to bioreactor, which is industrial scale. So a 50-liter bioreactor is typically what you need for a phase 1 clinical trial. A 500 liters bioreactor scale, typically when you 500 to 5,000, depend on the size of the clinical trial, you need again for phase 2 or phase 3 clinical trial. And you go to a much higher industrial scale bioreactor when you are looking into manufacturing and continuous supply into the market for patients post approval. So these are certain bioreactor levels once you are actually manufacturing a relevant biological compound. It is also important to understand that when you are looking into bioreactor processes, these are which we called by upstream bioprocessing. So these upstream bioprocessing, I'm using my laser pointer to understand. So this area where we are looking in upstream bioprocessing, these are extremely relevant to actually successfully launch your final material from a bioreactor. So once you understand that what kind of bioreactor I'm going to work on, what kind of upstream parameters I've decided, and if this understanding develops, largely you could actually reach to a very large level of bioreactor and go to a fill finish downstream level very effectively. Now, how do you do that? So this entire biomanufacturing area is largely operated based on experience. Currently, wherever the industry is operational, they are working on experience and trial and errors. And this is largely because of the bioprocess complexity. You are actually handling a living system. But there is a growing interest, and Industry 4 is coming on the digital twins. Digital twin models, which can integrate the physical and virtual system, again, by a real-time data monitoring. So you enable interactive communication, again, for advanced biomanufacturing and more operational efficiency. Digital twins, though it may be an alien name to some of you, but this has been operational for a very, very long time. Those who are in astronauts, those who are working in airline industry, they are using simulators before they are actually learning aeroplane, how to basically become a pilot or how to become an astronaut. You have to have into simulator. So these are a very simple concept which is there in the industry for a very long time. However, these concepts are very new to bioprocesses. Again, the primary limitation is complexity. And where is this complexity? Because of these factors which you are seeing on the slide. It means you have multiple process definitions which are typically needed in a bioreactor. A pH control, oxygen control, temperature control, pressure control. Amongst other controls, you have to always look into osmolality. You have to also have a cellular environment where cell growth, nutrients, product, and product quality are going to impact your bioreactor performance. Within the bioreactor, you have multiple components within the media like glucose, glutamine, which are also going to impact the performance of the protein production, together with process parameters, which are temperature, agitation, oxygen, or carbon dioxide. And which is also, the bioreactor is also going to run into batch, fed batch, and continuous operation. It means there are so many parameters which are actually needs to be understood while running a bioreactor, and that becomes one of the major challenge for developing certain computational models. So then digital twins comes with lot of hope to make these systems. So as I told you previously, these are virtual construct of physical system that mirror the behavior and dynamics of physical system. A complete digital twin must have a physical component. It means you need a bioreactor, actually a bioreactor and a virtual bioreactor. So there is a automated data communication. Virtual bioreactor, you can simply create one in computer, which will actually be correlated with your running bioreactor, and you can get the data transfer. Partial digital twins are also, which are actually being used where you collect the data, feed the data into computer and try to create. So this is happening for multiple years, but a real-time digital twin is still not a reality, which definitely companies who are developing bioreactor is a good motivation for them to go forward in this direction. Now, what are the existing computational approach to give you a closeness? DOE. So whenever I told you multiple data like critical quality attributes, key process parameters, so whether KPP, CPP, or CQA, those who are biopharmaceutical professional, they understand these terminologies very well. Design of experiment and multivariate data analysis, or MVDA, are again hands-on approaches. To minimize error, to have right scale-up options, people are utilizing design of experiment and multivariate data analysis for a while. DOE is a very is a historically, empirically established strategy. But eventually, mechanistic approach to understand mechanism, which are more computational in nature, and hybrid model, mechanistic and computational both, is also in right now. There are a lot of people which are going to, which are used in industry. CFD, or computational fluid dynamics, whenever flow pattern has to be decided, whenever you are developing a reactor and assuming a mixing dynamics, and you know how typically a fluid is mixed along with its component, and maybe if you are also designing how cell culture in a shear environment, or even other environment is how it is behaving together with bioreactor, with impeller and other bioreactor mechanics, you are actually deciding these things much before you actually do a experiment using CFD. CFD is again a age-old phenomena which is using. All these phenomena are not predictive in nature. So digital twin actually complement these methodologies for predictive behavior. So when you feed data, you are actually getting a more predictive behavior. You need much better control over your bioprocess before one can approach for scaling option from laboratory 1 liter scale to 20,000 liter scale. Maybe one can go directly to 20,000 scale if you have a real control over the data. So digital system, as I told you, physical system, which is a primary first need. What data needs to be captured here? So here, if you look into CPP, KPPs, which are your process parameters, your CQA, quality attributes of the design. Here you are running a CHO culture. Here the address which I am taking is for monoclonal antibody production. You are having multiple data cells, specifically temperature, DO, medium. They are actually controlling. So all these parameters are controlling your quality attributes, which will define your product quality, product yield, which also called specific productivity, aggregation of the protein, cell growth parameter. Still, we have yet to reach a real-time data calculation. So we have online analysis, inline analysis, in situ analysis, and these analyses are growing over the period of time. So we have reached to PAT and sensors to a where you could actually monitor DO, you could actually monitor temperature and pH in real time. But when it comes to quality attributes, only few quality attributes can be monitored inline. You need to capture data. So some of the data which can be captured in a bioreactor are actually very forward in terms of developing a model or a digital twin for that bioreactor for that particular situation. So cell culture data or real-time data monitoring of bioreactor for multiple set, which I have explained, and also the data which is continuously coming, how to process and how to manage. It is a large amount of data. So suppose you are running a parallel bioreactor, or you are running multiple bioreactors in sequence, or you are running different bioreactors for 13, 14 days culture, you are getting huge amount of data. So you should have a system how to manage. And current processing, digital processing, has this kind of challenge. But this challenge can be overcome by mechanistic and statistical model, which are already available, which are already being practiced for the cell culture as well as bioprocess development. So the virtual system, which is multi-scale mechanistic or data-driven, the kinetic model. So what are you are looking into? You are looking into different kind of approaches, which are cellular or reactor, either cellular approaches. So there are many genome scale model, which are there already established, and there are a couple of companies who are developing these models. There are a lot of other PTM, which are called post-translational modification. You have a kinetic model, which is based on entirely bioprocessing parameters. So this is already a multi-scale mechanistic model. These are small compartment. Since bioprocess is very complex, what people have done is compartment. So compartment modeling is developed in 1960s. Now the same compartment modeling you can actually apply in terms of developing bioprocess. You also have a data-driven statistical model, where you have multivariate data which is coming into statistical model, and through machine learning or neural network. And this is giving much more success in current model development. So these model which are coming and developing a digital twin are based on prior knowledge, historical data, or real-time collected data from the physical system. Now you are looking into a whole picture. How does this whole thing look like? So you have a bioreactor, since your cells are growing, which are going into different phases. Glucose level or amino acid level is also varied over the period of time when there is a growth in the cell, production in the phase. And the virtual system will also mimic the same situation. As I am saying, the physical system has to have a virtual system. So whether the virtual data, the way virtual data is going, whether you are getting same amino acid profile. So our group is continuously working on the same segment, specifically real-time monitoring of amino acid when we are running bioreactor, and real-time monitoring of these amino acids. We are also developing certain model in glycosylation, and specifically on charge variant analysis along with amino acid during these particular simulations, where we are feeding data from the real-time bioprocess to digital models, and whether the digital model are predicting that particular behavior, and whether other processes which are going to be follow-up processes can be developed much more effectively. In the data collection, that is one of the major challenge in upstream bioprocesses are there. So whether you have a small scale reactor or a bench or a pilot scale, you need these inline sensors. We also call them a lot of other sensors called soft sensors. Raman spectroscopy, which has, in recent time have proven its worth in a real-time understanding of multiple data. So what do you get? You get a reactor data like pH. You already have a lot of pH sensor. Cellular data like viable cell density, total cell density, glycan, metabolites, nutrients, amino acid. So these data is continuously coming when you are doing inline monitoring. And once your data processing is there, the Raman experiment for this particular, we have taken Raman, it goes to, again, an existing multivariate data analysis module, which give a PLS model deployment, and ultimately you get a process value. So once you run multiple bioprocess, you get a huge amount of data. As you see in other industries, big data analysis, data processing, data is oil, many other terminologies. Similarly, data is also becoming an important part of upstream. The primary challenge here is that industries, those who are actually working day in and day out on multiple bioprocesses, there is no control on that data which is coming out from. So it is largely going to be academically driven or driven by the startups or new companies who are doing data-driven model, and they are developing their bioprocesses. But the The success part will remain there when more and more bioprocesses data is uploaded on the system. So data processing can be much more smoother and finer. So I'd like to conclude here that physical and virtual bioreactors are interactively connected, and they can be connected by a management system of collected culture by inline monitoring. So in addition to bioreactor development, there are enough opportunities and future scope there where you could actually monitor the data which is coming out from the bioreactor. In virtual system, multiple development has happened on multi-scale mechanistic model or data-driven model. Data-driven is the future because it's easy compatibility with artificial intelligence and machine learning. And also they are combined to mathematically or statistically relevant input coordination. So like KPP and CPP, we all know what parameters are going to be input. We also know what are we needing as an output KPI or CQA. So since we are aware about input parameter and output responses, these model can simply facilitate the conversion. Fully integrated and again automated bioprocess digital twins where they can actually measure by soft sensors. These certain sensors actually are right now working on multiple. So a couple of industries are working for real-time culture profiling where they can also predict, and that is the true success of virtual or true success of digital twins, that they can understand metabolic changes, cell growth, and also the recombinant product synthesis. So the future which a digital twin will be going into, of course, AI/ML, which is also making a lot of sense in other industries, and the bioreactor companies should or must look into AI/ML integration in their bioreactor. Bioreactor simulator, which is also a couple of companies developing, but you need a more robust or harmonized simulator, relevant of the brands and complexities. Also, advanced control strategies where sensors are integrated. Almost all possible sensors one can make, specifically the chemical sensors. So pH and DO sensors are already there, but chemical sensor, there are a lot of development needs to be done. Digital biomanufacturing, through digital route, one can actually manufacture things on on computer, and can this be scaled up for much faster? And that is the one of the way to actually get affordability in biomanufacturing and impact the patient. So I wish to thank Archit and Marian, who in my team is working on digital twin model along with some of our collaborators. Thank you very much for your kind attention. And if you would like to learn about our work, please join on Biosimilar Workshop. Thank you very much. Over to you, Ashish.
Okay, sorry, I was on mute. So thank you, Dr. Ratnesh, for such a thoughtful presentation, which I'm sure has been filled with valuable insights for all the participants attending this webinar today. And we are already getting a lot of questions, which we'll take up in some time. Okay. And before we move further, we'll take up our next poll question. Request all our participants to kindly click the poll button. You will find the poll button after you click the Q&A button at the center of your screen. The second poll question is, what would be your current process scale? Option 1, lab scale, that is 1 to 10 liters. Option 2, process development scale, that is less than 100 liters. Option 3, production scale, that is more than 100 liters. We will allow 30 seconds for you to answer this poll. Thank you. And for our next session, I would like to invite Mr. Pankaj Salvi, Senior Bioreactor Specialist, Paul Corporation, and he will share his thoughts on the topic Stirred Tank Bioreactor, Benchtop to Manufacturing. Pankaj Salvi is a Senior Bioreactor Application Scientist and a member of the Cell Culture Technologies team at Paul Biotech. He has been dedicated to training bioreactor customers and providing scientific consultancy and advise on the transfer of their processes into Paul Bioreactor Technologies and process optimization. Pankaj holds a master degree in microbiology from University of Pune and has over 13 years of experience, including positions at the Lupin Limited and Paul. Previously, Pankaj has worked for Lupin Limited as an associate scientist process development and in a techno-commercial role in Paul. Knowledgeable and skilled in upstream and downstream process development, biolytics, and CGMP with experience in upstream and process development research on microbial and mammalian expression platforms. So over to you, Pankaj.
Thank you, Ashish, for the wonderful introduction. Good afternoon, everyone. So I'll be talking about benchtop to manufacturing. So essentially, like when a company plans to scale up from R&D to manufacturing, the major challenge that they face would be like how do they achieve their upstream scale up, and then again, like what are the what is the strategy to attain that scale up. So I'll be trying to to capture some inputs into how a company can achieve scale up and how can Paul assist you in doing that. So talking about the agenda, so the value proposition, I would be touching upon that, some from portfolio overview, some engineering and characterization aspects, scale up strategy, and a few case studies on that. Yeah, so the value proposition. So the Allegro STR bioreactor family is a single-use bioreactor, which is basically compact, which is essentially useful when in terms of usability. It's ergonomic that the users can easily handle it. It's intuitive, so that makes it easy for the user to reduce the risk of any mistakes. And then again, a turnkey solution, like it is a complete solution for you. And again, like process assurance and performance would be critical, basic requirement for a bioreactor. Talking about like cell lines which can be processed in Allegro STR. So there is a proof of like being used of SF9 to like NSO cells, even CHO cells, and like every cell line, basically a mammalian cell line can be grown in the system, which can be like suspension adapted, and sometimes like microcarrier based adherent cultures. So on the portfolio overview, this is basically based on customer feedback. I'll be starting with a video and I'll be talking through in that. So the 2000 liter bioreactor is basically a 3 feet bioreactor. So the 3 meters, sorry. So the 3 meter can easily fit into any facility. So you can see the packaging over here. It is very compact and it is easy for 2 personnel to install it. And then again, there are like interlocks which would guide you. And there are color coded tubing labels. This is the intuitive screen. And it is an automated inflation. So essentially there is no operator intervention during inflation. And then there is automated deflation, which would reduce your disposal volume. Yeah. And then again, like when you're going to pick it up, it is just like 2 personnel required because of the excellent packaging. And then talking about the performance, like there is excellent KLA, greater than 30 KLA. And then cell growth and viability, which I would be touching upon in my case studies. Yeah. So talking about the key differentiators, like how does Paul Bioreactor stand? So usability wise, like the installation is pretty easy. Only 2 operators are required for the installation and complete operations, and like a reduced footprint. Process assurance wise, like it is like 100% integrity tested. So and validate like the bio container is 100% integrity tested. That gives you the assurance over failures of the SUS. Again, like validated software is in there. And talking about performance, like we have a large pitch blade impeller, which basically delivers a high power input, and that will help you to get a good KLA. That I would be touching upon in the case studies. Talking about the portfolio, like Paul has invested a lot in the Allegro STRs, and you can see like in 2014, Paul started with a 2200 liter bioreactor. And then now by 2020, we have the complete range starting from like 50 to 2000. And talking about the engineering and characterization performance aspects. So this is the feedback from the customers, what the voice of the customer, and this is not what Paul says. So in terms of ergonomics, it is very compactly packed, so which helps you in handling. So handling is very easy, and like two personnel can handle it. Installation wise, the installation feature is like the open face structure of the bioreactor being a cubicle helps you to install it easily. You can just front load the bio container. Installation wise, it will just take 10 minutes to install it. And then talking about automated inflation, it takes like 30 minutes against a cylindrical bioreactor, which can take about 90 minutes for a 2000 liters. The tubings are color coded and numerical. So the coding helps you to install it. It is very intuitive. Talking about the footprint, the 2000 liter bioreactor is just like 3 meters, which because of which you don't need a ladder, only a step stool can work. Talking about manpower like two personnel can handle the entire setup. And automated deflation, which reduces your disposal volume. So that basically gives you an assurance that there would be reduced errors in the manufacturing. Talking about process assurance differentiators. So like the differentiation factor is the packaging. The packaging is very compact and neatly planned, which makes it much easy to handle, and then it reduces the chances of any damage to the SUS. There's a bottom entry impeller which has a short shaft, which essentially would reduce the stress on the bearings, and that would like reduce your chances of any leakages. The tubing management is smartly done, which like during inflation, you can easily like without any operator intervention, the tubing would align properly. Handles, there are bioreactor handles for like loading the bio container, and then there are rip cords which avoid any sharp edges. Talking about the sensors, the process control. So there are like single use as well as conventional sensor options for the pH and DO. And then again, like based on the process requirement, the DCO2 sensors or any other pad tool can be incorporated into the sensor system. Talking about like engineering performance and mixing efficiency. So based on like when it was in the R&D development, so based on the CFD model, a pitch blade impeller was possible because of a direct drive impeller. And this essentially has given the Paul the flexibility of having a big impeller that is like half the diameter of the bioreactor, which helps to drive better power input. And with this, you can actually get a KLA more than 30 per hour, which essentially gives you like the confidence that you can work in a big process range for the power input and get your process scaled up, and essentially a good KLA. Talking about the engineering performance in terms of mixing time, which becomes very critical in terms of a bioreactor. So this study was done with a media simulant. So the media simulant was used, and the mixing time was evaluated with a addition of a alkali based on the pH to achieve like 95% of your fixed set point, stable set point. So essentially you can see over here that the graph shows that each of the system is basically scalable and it's similar in performance. And then with like high power input, you can even achieve a mixing time of less than 30 seconds and even like less than 15 seconds. Like KLA, like oxygen transfer and KLA. Though these are the important criteria when someone is evaluating a bioreactor and when you're trying to achieve good performance in a bioreactor. So talking about KLA, like you can see like with a 1000 liter bioreactor, we can touch the range of KLA in this range of 40 per hour. And essentially like every bioreactor can achieve around 30 KLA. And again, like if you see this graph, the predicted KLA versus measured KLA. So the predicted KLA is basically based on the Van T rate equation, which is basically dependent on the power input per volume and the superficial gas velocity. So in this case, you can see that the predicted and the measured KLA are similar. So basically this indicates that if you're going to scale up a process based on only the power input and the superficial gas velocity, you can easily achieve your targeted performance without actually doing a KLA study also. So Paul can help you with that factor. And yeah, talking about like CO2 evacuation, like essentially when someone is scaling it up like to a 1000 liter scale or a 2000 liter scale, one critical factor comes into picture is the CO2 accumulation in the system, like in the media. So the dissolved CO2, if it increases, it is going to be toxic to the cells. And considering this, the essential thing that we need to check is like the stripping out of the CO2. So for this, the study you can see like with the Paul Allegro STR, which has a macro sparger, like with even like we can give in higher sparging of air, and this essentially helps you to achieve good CO2 stripping, like it is like in the range of more than 1 mole per liter per day. And like this was evaluated against an in-house perfusion process where you get like 146 million cells, and again with a fed batch process with 23 million cells. Yeah, and talking about like computational fluid dynamic modeling. So this study was essentially done for a comparison between the cubical bioreactor and the cylindrical bioreactor. So this with this graph, you can essentially see like with the Kolmogorov length between the two bioreactors is basically similar, and again like the power input per volume is similar between the two bioreactors, and this image basically shows that there is essentially no difference between a cubical Allegro STR and a standard cylindrical bioreactor. And yeah, like how can we help you with the scale up? So scale up basically like with literature you can understand that scale up is basically a function of your mixing and the mass transfer. If you can achieve these two factors, you can essentially scale a process well. So these being the nonlinear scale up parameters. So basically it has been seen that if you can match the power input per volume and the superficial gas velocity, you can have good mixing and mass transfer, and it is irrespective of any bioreactor size. It can be a benchtop bioreactor or a 2000 liter bioreactor. So essentially what Paul does is like we would be with the extensive study available, we can help you with the achieving a constant P by V and superficial gas velocity. And with a good PID control, you can have good environmental control and gassing in the system. So this is an example, like usually like how do we go around with scale up? So over here you can see like this scale up, it was done from a 1.5 liter traditional 1.4 liter traditional bioreactor and against a 2000 liter Allegro STR 200 liter. And you can see over here, like essentially the power input of like 70 was achieved. But if you keep a constant PYE, you can achieve that same in the Allegro STR. Whereas if you consider a tip speed, in that case, like you would have a lower KLA. Over here only, if you can see over here, it's just 2 against 9. Having a constant power input and a superficial gas velocity would essentially help you to scale the process in the right way. And then again, like if you consider VVM as a scale up parameter, then you would be adding excess gas into the system, which is not required, and that would reduce your performance. Talking about scale up strategy, how we can help you with that. So some bioreactor application specialist, trained bioreactor application specialist can help you with that scale up. So we would be assisting you with the scale up. So for that, we would essentially require some inputs from your end. That is the current reactor specifications, what are the impeller specification and sparger specifications, which can be obtained from the vendor or the bench scale bioreactor vendor, or essentially from data sheets. And then based on that, we can help you achieve that scale up. Now, yeah, I'll be touching upon a case study. So this case study is basically on AAV production in a stable transfected cell line. And this basically is public literature. So this is this is from CEVIC. And over here, you can see like an immortalized human aminocyte cell line was selected. And it was a stably transfected cell line producing AAVs. So basically the process which Paul got was in a 10 liter stirred tank bioreactor, benchtop reactor. And then the criteria was like we need to scale it up to a 50 liters and a 200 liters. So the strategy employed over here was essentially getting a fixed power input per volume and then maintaining the superficial gas velocity in there. So this is the scale up tool that we employ, in which we would need some inputs from the customer. That is basically getting the geometry of the system, getting the inputs on the agitator, basically the power number and how it is operated in upflow or downflow. And again, like how is the sparger? Is it a micro sparger? Is it a macro sparger? So essentially, even if the benchtop bioreactor is a micro sparger, we can scale it up in an Allegro STR in a macro sparger way. And with these inputs, and then additional inputs like inputs with the present process with the agitation speed and the sparger flow, we can achieve that constant power input over here, as you can see. And the superficial gas velocity, which essentially would reflect in your KLA. And like the scale up study further on, like if you have those inputs, we can even evaluate further parameters like Kolmogorov length, the tip speed, the average shear rate, and then mixing time. This would be critical in sense like if the customer is very cautious about like with very sensitive cell lines where the shear can be pretty high. Shear can create a damage to the cell. Talking about the performance, so this is the study where essentially this line indicates, the purple line indicates the Allegro, the BioBlue bioreactor 10 liter scale, and the other lines indicate the Allegro STR50 and the Allegro STR200. And over here you can see like if not like you get comparative growth achieved in this bioreactor and viability, and essentially I would say better viability and growth. And then same compared to in terms of titers, we've got comparable titers in both the systems, in the Benchtop bioreactor as well as the scaled up bioreactor in the Allegro STR. Another case study touching upon like a comparison between the cubical traditional cylindrical bioreactor and the cubical bioreactor Allegro STR. So over here essentially we have seen like maintaining the same power input. Basically we can achieve a scale up, or I would say essentially like the same conditions between a cylindrical bioreactor and a Allegro STR cubical bioreactor. So in this case all the parameters like linear parameters work maintain the same, and the power input basically was maintained the same with the differences in the agitation. So this was a cell line, CHO S cell line, with in which the product was the IgG1, and we have this as a published literature which can be available to the customer. And talking about the performance, so essentially like similar or even better performance was obtained in the Allegro STR against a cylindrical 200 liter bioreactor. Over here, this indicates the Allegro STR, whereas this indicates the cylindrical bioreactor. And again, in terms of titers also, better titers or comparative titers were achieved in the Allegro STR against a cylindrical bioreactor. Talking about the case study, another case study. This is about like SF9, and this is again the published literature. This was like published by Brahma, and Brahma. This was a study of SF9 cells, which were co-infected with baculovirus 2 baculovirus vectors to produce an AAV or GF vector. And this essentially indicates like the data is available to be shared, like it is a published literature, so that can be shared. And you can see the titers obtained in a Paul STR bioreactor against a cylindrical 200 liter bioreactor, and essentially they are comparable. Talking about the support services what we can provide, so we have application scientists which would be there to train the customer and even support you with consultation and process support with troubleshooting as well. There is service available from our side, like whenever is required, there are trained service personnel available. Validation services are available for single use when it comes to like extractable leachables and other aspects of single use. And then process development services are also available from Paul. And it being a global service network, you can assure you that we can provide you the most efficient support. And yeah, talking about the seed train, so we have an XRS bioreactor which can support you with this is a rocking motion bioreactor which can support you with your seed train requirements. So I'll just run you through the video. This is essentially a biaxial rocking motion. So instead of a single axis, it is like a biaxial mixing, which gives you better mass transfer and better mixing in the system. So essentially you can obtain a better KLA than a traditional single axis rocking motion bioreactor. And yes, like support wise, Paul can support you end to end. So they can provide you end to end solutions, not only the bioreactor, but with your downstream solutions, right from clarification to your fill finish operations. Thank you.
Thank you for that insightful session, Pankaj. And now it's time for our final poll question. Request you all to kindly click the poll button. And the next question is, are you interested to get a technical support from Paul for your upstream process? Option 1, yes, please contact me. And option 2, no, we can do it later. So we'll spend another 30 seconds to make sure that we answer this question. Okay, now I would like to open the floor for the participants to ask questions they would like our speakers to share their views or perspectives based on the experience and expertise. You can also join the stage and ask questions to our speakers one-on-one. You just have to click join stage button and just write your name and you can log in. In the meantime, let me get all our speakers on the floor. Welcome everyone, and thank you all for wonderful presentations. As you see that we have a lot of questions already, and we'll go one by one. So the first question goes for Mr. Satish. How do you maintain temperature for SBUs? How can we recycle the SBU when it is categorized in the biohazard waste?
Yeah. There are already well-established heating pads are available for heating single-use reactors. There is no issue. And as far as recycling is concerned, so you can initially decontaminate the single-use reactor, then you can recycle it. You can validate that process. I think this answers your question.
Okay. There is one more question for Mr. Pankaj. Which engineering parameter should be critical during process scale-up from cylindrical geometry to cubical geometry?
Yeah, so hi. So I guess I already answered that in my presentation. So that wouldn't differ actually, because a cubical and a cylindrical essentially are the same, because if you've seen, like, a cylindrical would always have baffles. So if you see internally a cylindrical bioreactor, it would be similar to a cubical being a natural baffling system. And additionally, like, the parameters don't vary when you're going to scale it up. It would be essentially the same. Like, you can consider KLA as a primary factor, the mixing time, and then KLA would be like a predictive model would be your superficial gas velocity and your power input per volume.
Okay.
And apart from that, there could be some other parameters which we need to work on. Like, yeah.
Thank you. Our next question is for Dr. Jain. We are working on a metabolite using recombinant E. coli. Our question is for large-scale bacterial culture. If we want to change the defined media in between the experiment, for example, supplement of substrates, can we able to install a microfilter?
Yeah, so thank you for this question. First of all, with respect to large scale, large scale culture, whenever one is conducting, it is not advised to change media. When you reach to the media stage, when large scale culture is going on, it assumes that the media conditions are being optimized. So it is not advised that one is actually one is changing that stage. As long as filtration systems is concerned, I'll probably pass this question to Pankaj. Probably he could answer that question in a more, much better way. So Pankaj or Satish, both of them can probably answer whether a microfilter attachment may be a nice idea or not.
Yeah, any of you who can pick?
Yeah. So essentially what I understand is like it was like hollow fibers or something that has been asked over here. So I've seen that it has been used a lot, like hollow fiber based ATF systems, or you can consider hollow fiber systems like APS have been used in mammalian cell cultures. But bacterially, essentially it has like, there is less application in that sense, except for like used for like the final processing steps where they need to remove the IBs, separate the IBs. So essentially, I wouldn't say like this is not a common practice to change the media in microbial processes, because again, like microbial processes are very dynamic, and it is like there is a rapid change. So changing a media would be like difficult, and again, bacteria would tend to clog the filters, the hollow fibers or something. So it would require a lot of optimization, but that would again not be economical, considering like E. coli would be a process where you would consider economics in that process. I think, yeah, that's it from me.
Okay. Anything from your side, Satish? Satish, you're on mute. I believe there's some issue with his network. Okay, now we have few people who have joined the stage, and I'll take Mr. Siddharaju to ask a question here. Siddharaju, can you please unmute yourself and you can ask a question?
Ashish, I have a question for
and I'll take
Pankaj.
Mr. Siddharaju.
Yeah, with respect to KLA calculations, Pankaj.
Siddharaju, can you please unmute yourself and you can ask a question?
Yeah, I have unmuted.
I have a question for Siddharaju. You need to put yourself your front end on mute. Siddharaju, can you please unmute yourself and you can ask a question?
Yeah, I have unmuted. I have a question for
Siddharaju. You need to put yourself your front end on mute. Okay, let me inform everyone who is joining on the backstage. They are also logged in at the front end, so there will be an echo. So if you are joining backstage, please switch, or maybe you can mute your front end so there's no echo. Siddharaju, can I? No worries, Siddharaju. Do you want to join again? You can just mute yourself on the front end. Can I take you on stage? Can you ask the question, Siddharaju? You can unmute yourself.
And he's written that he'll Pankaj after the call maybe.
I think it's all messed up here. Like maybe.
Yeah, in case like if someone wants to connect with us later, we can do that also. We can answer the questions in separately. Yeah.
Sure, no worries. We have a lot more questions coming up here. Max scale of SUB in the market today and challenges encountered during implementation. This is a common question. I think anyone can pick this up.
So I will comment on it probably. Like single-use bioreactors, considering stirred tank bioreactors, the max scale I would say like which is there in the market now is like I would say like 6,000 liters. Like less proven, but yeah, there is 6,000 liter scale which I have seen. There is 4K, 5K, and 6K from different vendors. And the challenges, I would say, like as you increase the scale, like the chances of leakages in like would increase essentially. So because of the hydrostatic pressure building in and then getting the right, like the leakages would be a primary concern, I would say, in that case. And again, like it depends on the technology, like vendor to vendor. It would be like meeting the heating requirements. Your gassing requirements may also be a challenge in that sense in a single use.
Yeah. So I think Pankaj has rightly said that 4 to 6 KL scale, but 4 KL scale is something which is very well proven in terms of single-use bioreactors. In most of so it doesn't matter what kind of vendor system you are choosing in, but 4 KL scale is very well proven and that has demonstrated its success. As rightly pointed out again by him, that if you go on a higher scale, there are companies, major contract research organizations, specifically in out of India, like Samsung, which have tried certain other scales as well on a higher scale. But largely India or in Indian perspectives, 4KL is a very well accepted scale, and also current, considering the need, rarely one has to go beyond that scale as well, unless the COVID vaccination kind of a situation comes in. But for routine antibody manufacturing or routine enzyme manufacturing as well, one can definitely manage at 4KL scale, and that is also economically much more sensible. Yeah.
Thank you. Before we move ahead, one more question for Satish. We are just running out of time. We'll just take one question each now. Microcarrier suspension based Cytodex or fixed bed par iCellus? Which platform is better for adherent cell culture? Satish, this question is for you. Satish, can you hear us? I believe he has some issue with the network. Pankaj or Ratnesh, can you pick up the question here?
So
Yes.
essentially, like, yeah, sir, would you like to go
no,
ahead?
Pankaj, please go on.
Yeah.
I'll add on.
Yeah, sure.
Please
So
go on.
essentially, like talking about like a fixed bed bioreactor, like something like iCellus or a microcarrier, I wouldn't say like anything is better. Like it is depends on your culture conditions also, like how is your cell line and all that. So because microcarriers would be in a stirred tank bioreactor, whereas a fixed bed bioreactor, like essentially like this would be an adherent system which is fixed, and then the flow would be isolated, like the stirring would be isolated. So being two different systems, again it needs to be evaluated for your system individually. And talking about like stirred tank bioreactors, what I have seen is like achieving a scale up beyond 500 liters is sometimes a challenge, especially when you're talking about like gene therapy applications, where getting the transfection becomes a little complicated at like higher scales. Whereas in adherent system, like you have like around 500 meters square, like for iCellus, where it doesn't become a challenge, at least achieving the transfection. So yeah, again, but it depends on your basically cell line and what process you're running. And I would say like it essentially needs to be evaluated in both cases.
Dr. Jain, do you want to add something?
Yes, am I audible?
Yes, you are.
Yes. So I think I'll just, in agreement, there is no priority, but one of the major thing one has to take between iCellus or microcarrier based culture is when you are looking for density. If your aim is to achieve highest possible density, iCellus has certain proven advantages you could achieve through microcarriers as well. And microcarrier has provided a good amount of productivity as well as achieving a cell density. Since because of the nanofibrous technology, because of the inherent advantage of the technology, iCellus has given certain advantage. So if your objective is to achieve higher cell density, then of course you could probably prefer that. Though again, there is no preference over. So what is your objective? That makes then you can choose. But if you are just exploring, you can begin in both the cases. It doesn't matter what kind of technology you are choosing.
Thank you. The next question is for Pankaj. All bioreactors is cubical, then doesn't it differ in mass transfer and performance from a traditional cylindrical bioreactor?
Yeah, so like again, I guess that this is a similar question to the prior question. So yeah, though it is a cubical thing, like basically like what was the reason for a bioreactor being cylindrical was like earlier vessels were all SS, and SS systems need to be like sterilized. They were like SIP. So SIP basically are pressure vessels. So because of that primary reason, a vessel being SIP, it needed to be a pressure vessel, which is basically a cylindrical thing. But when we think about single use, that because of the irradiation you don't essentially need to SIP it. So that like eliminates the reason for keeping it a cylindrical, because even in a cylindrical bioreactor, if you see, like you're not going to have a complete cylindrical inner surface, you're going to have baffles in there. You need to restrict that like mass movement of liquid. So that essentially a cubical bioreactor would be a natural baffling system, which would get you will enable you to achieve the same thing. So essentially I wouldn't say like when it depends on the need, but again, like there is not much of a difference, and that has been like even with my data, if you can see, they are like performing in similar ways, in fact.
Okay, thank you very much. We'll take one last question. Okay, we have a question from the, just give me a second, I'll pick the question. Okay, how do we do risk assessment during bioreactions, and how do we develop control strategies as a part of QBD? Satish, would like to take this up? Are you able to hear us?
Hi, hello. Can you repeat
Yes,
the
we
question?
can hear you. Yeah, how do we do risk assessment during bioreactions, and how do we develop control strategies as a part of QBD?
There are various tools available as far as risk assessment is concerned. So you can do the risk. There is no difference between the risk assessment as far as conventional or single-use bioreactor. The risk assessment remains same. So you can use the same tools conventionally what we are using. So same way you can perform risk assessment. As far as QBD is concerned, same thing. You can perform the risk assessment on the that aspect also. Doesn't differentiate between conventional as well as single use. It remains same. So maybe a number of items which you cover may be reduced, but the tools remain same. You can use the same old tools for risk assessment as well as QBD. I think that answers your question.
Great. Thank you very much. I believe we have already run out of our time, so we will stop here. I would like to thank all the audience for being such a wonderful audience here, and I would also like to thank and take this opportunity our esteemed speakers for taking out time from their busy schedule to be a part of this exclusive webinar, which surely has been engaging and enriching for all our participants. I would request all our participants to click the feedback button and take a time to give your valuable feedback. This will help us to serve you better. You will find this feedback button on your screen. Just click the Q&A and you will see the pop-up of feedback button. Just take a minute and you will be able to give your feedback. Also, don't forget, whoever has not filled the poll questions, it's still active. Click the poll button, submit your answers before you leave. So once again, thank you for being such a wonderful audience. With that, we conclude this webinar on Upstream Bioreactor Technology, Benchtop to Manufacturing, brought to you by BlueTech Media and powered by Paul Corporation. But having said that, this is not the end. We will be coming up with our next session that will be talking about the adherent cell culture webinar this month itself on 28th of April. So stay tuned and we will connect again. Thank you very much. Stay safe.